The second part of the cytoskeleton is the microfilament. Microfilaments also have a repeating monomer subunit, the protein actin. Unlike microtubules, which are actually hollow tubes, microfilaments are simply long ropes of actin. Now, when we talk about actin in regards to microfilaments, we talk about the individual actin monomers as either G-actin or F-actin. And by using those names, we're referring to an individual actin molecule as being globular, meaning it has not yet been attached to the microfilament, or being filamentous, meaning it has already been polymerized. The microfilament is actually composed from two strands of actin molecules all joined together, and those two strands are then wound around each other. On this slide, you can see a drawing of a microfilament, and you can also see an image taken under an electron microscope showing a tangle of microfilaments in a cell. The microfilament is made out of two strands of actin molecules all joined together, and those two strands are then wound around each other. Microfilaments are a much smaller structure than microtubules are. Remember, we said microtubules have a diameter of about 25 nanometers. The microfilament is only about 7 nanometers across, so much, much smaller. The actin molecules shown as these golden yellow circles, they are referred to as G-actin when they are free floating in the cell and not yet attached to the microfilament. Once they are part of the microfilament, they're referred to as F-actin. What you're looking at on the bottom here is another view of a microfilament. The two strands are depicted in blue and white. And what this diagram reminds us is that each one of these golden yellow circles is a protein. It's a protein that is made out of hundreds or thousands of amino acids. So all of the little tiny circles that you can see in white and in blue represent the individual amino acids that make up each one of these actin monomers. Now I want you to notice in the electron microscope image over here on the right how tangled these microfilaments look. It's very different from the image and the video we looked at regarding microtubule structure. Microfilaments are used for a different purpose than microtubules are, and so they're going to have a different appearance in the cell. Just as we saw with the microtubules, microfilaments have directionality. One end of the microfilament is going to look different than the other end. We refer to the plus and minus ends of a microfilament just like we do with microtubules, but we also use the terms pointed and barbed. So the plus end is referred to as the barbed end, and the minus end is referred to as the pointed end. And that's because when you look at F-actin, when you look at actin, once it becomes part of a microfilament, it takes on a shape something like an arrowhead. And of course, this arrow has a pointed side and a barbed side. So when you put lots and lots of filamentous actin together, it means that on one end will be the point, and on the other end will be the barb. So the plus end of a microfilament is also the barbed end. And the minus end of a microfilament is also the pointed end. Remember, there are three basic functions that the cytoskeleton has. It maintains cell shape, it moves things around within the cell, and it helps move the cell itself through its environment. We looked at all of the ways that microtubules participate in all three of those functions. And as you can see on this slide, microfilaments also participate in all three of those functions. They help maintain cell shape, they help move cell contents around, and they also help move cells around. When it comes to maintaining cell shape, what microfilaments do primarily is provide structural support underneath the plasma membrane. They make up, for example, the very core of a microvillus or any kind of membrane extension that comes off of a cell. And we'll look at a picture of this in just a moment. When it comes to moving things around in a cell, microfilaments participate in a process called cytoplasmic streaming. Just as blood circulates through your blood vessels, inside of each one of your cells, the cytoplasm circulates around the cell as well, and microfilaments help facilitate that. During cell division, when the cell is ready to go through cytokinesis, through the actual division of the parent cell into two daughter cells, it's microfilaments that facilitate that. They arrange themselves around the periphery of the cell in the center, and then they contract inwards, causing the cell to physically split in half. When it comes to the movement of cells, microfilaments actually participate in what we would think of as amoeboid movement, in the kind of movement that involves membrane extensions, reaching out and sort of pulling a cell along through its environment, the way amoeba move. It's the microfilaments that help form the pseudopods, the philopods, the lamellopods, all of the different varieties of membrane extensions that cells use to move themselves through their environment sometimes. The other type of cellular movement that microfilaments are involved in is contraction and relaxation. This is the same process that your muscle cells use, the process of contracting or shortening in their length, and then relaxation, extending in their length. And microfilaments facilitate that process as well. On this slide, we're looking at some diagrams of microfilaments alongside some EM images. If we start with the image on the left, this is a drawing of a single microvillus, the kind of finger-like membrane extension that you would see, say, in the intestine. Remember, each intestinal cell sends out these microvilli on their apical surface on the side of the cell that faces the cavity of the intestine. And the purpose of these extensions, the purpose of these microvilli, is to allow for an increase in surface area for each cell so that an increased absorption can occur. Now, inside each microvillus are these linear support structures. These are microfilaments. They are literally giving the microvillus its structure. Over here is the EM image. We're looking at a single intestinal cell, in fact, only a small region in that single intestinal cell. This is the apical surface. This is the side of the cell that faces the cavity of the intestine. And you can see that it's not just a straight surface. It's been shaped into these finger-like projections that we call microvilli. At the core of each microvillus are microfilaments. In fact, you can see how the microfilaments actually extend down into the rest of the cell, giving sort of a base to this structure and helping it keep its shape. Over here on the right at the top, you're looking at a piece of plasma membrane. The inside of the cell is shown in yellow. The outside of the cell is shown in blue. You can see the individual phospholipids here. You can see in purple various proteins inserted in the membrane. What I want you to notice is this down here. There are microfilaments arranged underneath the plasma membrane on the cytoplasmic side. And these microfilaments, along with lots of accessory proteins helping to hold them in place, these microfilaments help maintain the shape of the cell by arranging themselves underneath the plasma membrane. What you're looking at here is the surface, the undersurface of a portion of a plasma membrane. And as you can see, there is this very geometric looking arrangement underneath the plasma membrane. These are microfilaments, helping to maintain the shape of the cell. And as we said, microfilaments are also involved in helping cells move through their environment using this amoeboid movement. So primitive eukaryotic organisms like the tetrahymena that we looked at in lab can sometimes form these membrane extensions, pseudopods and philopods and lamellipods, and they're able to do that because of these blue lines in here, which represent microfilaments. Every time the cell rearranges the microfilaments, it can send out an extension of the membrane and then pull the cell along through the environment. What you're looking at on the bottom part of the drawing is simply the different ways that microfilaments can be arranged, depending on the type of extension the cell is going to use. Microfilaments and microtubules have a lot of features in common. It turns out that microfilaments treadmill the same way that microtubules do. There will be an ATP actin cap on the plus end of a microfilament. Now, microfilaments use ATP instead of GTP as the energy-rich molecule that helps facilitate polymerization, but otherwise the process is very similar. The ATP actin joins onto the microfilament at the plus end, creating this cap, and the cap attracts more ATP actin. The actin molecules can be removed from the minus end, and if the rate of adding new actin to the plus end is the same as the rate of removing actin from the minus end, then the microfilament treadmills the same way a microtubule does. One thing that's different between the microtubule and the microfilament is that microfilaments have an amazing array of accessory proteins that tend to associate with them in the cell. I've got a list of them here. There are what we refer to as cap proteins, cross-link proteins, severing proteins, bundling proteins, branching proteins. All of these accessory proteins are going to allow microfilaments to take on a much greater array of assemblage inside the cell. In other words, they're going to help create that sort of mesh-like look that we saw in the EM image earlier in the lecture. Take a look at the EM image on this slide over on the left. This is an array of microfilaments. Now, it's been colorized. They're not really green and yellow and blue, but they're showing us that microfilaments are not just linear structures the way we tend to think of microtubules. Microfilaments are commonly branched. So what you're looking at on the right-hand side is a depiction of some of these accessory proteins, allowing for this more mesh-like arrangement of microfilaments. So there are specific names of proteins listed in this diagram. Please don't worry about those. Just understand that there are certain proteins that help microfilaments branch. There are other proteins that will cap the ends of microfilaments sometimes so they can no longer shrink down or grow. There are some that help sever the microfilaments at a particular length. There are all kinds of accessory proteins involved in the arrangement of microfilaments. This slide is also showing some of these different arrangements. In the center, from this area over here on the left inwards, we're looking at how a microfilament is originally assembled. The G-actin monomers will attach to ATP molecules, and they will come in and they will be arranged into the microfilament. They will polymerize into the microfilament. Once the microfilament is formed and it's treadmilling, these accessory proteins can start to alter the architecture of how these microfilaments sit in the cell. You can see some bundling together that happens with bundling proteins. You can see some severing going on and some capping, as I said earlier. You can have cross-linking happening. You can have anchoring proteins attach microfilaments to the membrane. There are all different arrangements that these microfilaments can take in the cell because of the actions of these accessory proteins. Now, the last component of the cytoskeleton is the intermediate filament. It got its name because in size it falls between the microtubule and the microfilament. Remember, the microtubule has a diameter of about 25 nanometers, the microfilament about 7 nanometers. Intermediate filaments vary a little bit, but generally between about 8 and 12 nanometers. So they got this name intermediate because they're intermediate in size between the other two. Now, intermediate filaments are quite different from microtubules and microfilaments, most notably because they are composed of different protein subunits in different cells. In other words, the intermediate filaments that you would find in a neuron are going to look very different from the intermediate filaments that you would find in a skin cell. The protein that makes up the intermediate filaments is cell specific. And of course, there are many different proteins because there are many different cells. We actually divide all of the different proteins that can make an intermediate filament into these big classes. There are 6 of them. So lots and lots of different proteins can make up an intermediate filament. It all depends upon the cell type. Another big difference between intermediate filaments and the other components of the cytoskeleton is that intermediate filaments have no directionality and they do not treadmill. So they are a very stable structure. Once the cell creates the intermediate filament, it is created. It's not going to be dynamically altered in its length. It will have a certain lifespan in the cell, and then it will be broken down and recycled. When we look at the structure of intermediate filaments, they actually have what we call a cable structure. Now, if you're not familiar with that word cable, it actually has a definition. It actually involves not just two linear structures winding around each other, but multiple combinations of those two linear structures. So I'll show you a picture of that in just a minute. So identical ends on each end of an intermediate filament, no directionality, no treadmilling, so they are the most stable component of the cytoskeleton. I included this table from a textbook just to show you these classes of protein types that we know can build intermediate filaments inside of cells. There's nothing for you to learn in terms of the names of these proteins. I just want you to be able to see that there is a variety of them by cell type. So, for example, there's a type of protein called vimentin that we find in class number 3. And this particular protein is going to build intermediate filaments in fibroblasts, which are one of the components of connective tissue. There are other proteins called lamin proteins that are going to help to build the nuclear lamina, which we find in all types of cells. So lots and lots of different proteins that can be broken down into these 6 classes, all capable of making intermediate filaments inside of different cell types. On this slide, we can see a little bit more in terms of the structural detail of intermediate filaments. The way these filaments are assembled involves first the individual proteins involved dimerizing and then forming what we call tetramers. So if you look over in the diagram on the right side, this right here represents a dimer. So they're showing us 2 proteins, one in this white color and one in the gold color. And they're showing us how the proteins have their N terminus and their C terminus. Remember what that means. So these 2 proteins, and you can see they have a very elongated shape to them. They're going to wind around each other and create a dimer. Now, 2 dimers are then going to come together like this and form a tetramer, with tetra meaning 4. Then the tetramers are going to assemble themselves in this staggered arrangement, creating a protofilament. And then the protofilaments will join together to form the intermediate filament. This is a very complex arrangement, and this is why we refer to this as a cable. Now, in terms of the functions that intermediate filaments participate in, they are primarily involved in maintaining cell shape. Remember, we said there are 3 jobs that the cytoskeleton performs. It maintains cell shape, it moves things around within the cell, and it helps move the cell itself. Microtubules and microfilaments participate in all 3 of those functions. But intermediate filaments are different. Intermediate filaments are only going to participate in maintaining cell shape. So they are part of the network of cytoskeleton that forms the basic scaffolding within a cell. Now, one of the notable things about intermediate filaments is that they are very strong, very resilient. And while all 3 components of the cytoskeleton can resist certain forces that are placed on a cell, it's the intermediate filaments that are best at that. They can resist pulling forces and pushing forces and torquing forces and help that little cell maintain its shape. The intermediate filaments are also involved in anchoring certain organelles in place. Remember, some organelles are quite mobile in the cell, but other organelles tend to sit still, including, most notably, the nucleus. So intermediate filaments are helping to maintain the cell's shape, and they are giving the cell resilience against the forces that might impact that shape. Another thing that's important to remember about intermediate filaments is that while they don't treadmill, that does not mean that they are completely static structures. Intermediate filaments, like all components of the cytoskeleton, have to be moved around in the cell. They have to be created at certain times, destroyed at certain times. It's important that they can be dynamically used during the lifetime of a cell according to its needs. So while the actual intermediate filaments are not very dynamic, they are not sitting still in a cell. They're being transported around and used according to the needs of the cell. A great example of this is the nuclear lamins, again, that are sitting underneath the nuclear envelope. During the process of cell division, during mitosis, if you can recall from your earlier courses, the nuclear membrane has to break down so that all of the chromosomes can be released into the cytoplasm. Remember, we just said that nuclear lamins are the kinds of proteins that are used to build intermediate filaments, and that's exactly what the lamina underneath the nuclear envelope is. It's made out of intermediate filaments. So during the process of cell division, during mitosis specifically, that structure, that nuclear lamina, has to be disassembled. And the way the cell does that is by phosphorylating the proteins, and it helps those proteins change shape and then disassemble. So the nuclear envelope breaks down during mitosis and then is reformed again once the two daughter cells are created. So to conclude today, the cytoskeleton is a structure inside of living eukaryotic cells that we often overlook because we don't tend to see it in the kinds of microscopic images that we're used to viewing. The cytoskeleton requires special staining techniques to be seen, and components of it cannot be seen under a light microscope. You have to look under an electron microscope. So we have to remind ourselves of the cytoskeleton and the important work that it's doing. It is a very dynamic structure. It is tightly regulated and constantly changing in eukaryotic cells. The microtubules, the microfilaments, and the intermediate filaments that make up the cytoskeleton each play critical roles in the cell. And each of those three structural components of the cytoskeleton are integrated with each other. They play different roles, but those different roles are going to work together to allow the cytoskeleton to complete its three important jobs: maintaining the shape of the cell, moving things around in the cell, and moving the cell itself. For example, when it comes to maintaining cell shape, we said the microtubules are very good at resisting the compressive forces that are sometimes applied to a cell, those sort of pushing forces. The microfilaments are very good at generating tension to move a cell in terms of contracting and relaxing. The intermediate filaments provide a whole lot of protection against unusual forces, torquing forces. All three of those things are happening at any one time, and all three of those things are important for how a cell maintains its shape while still being able to perform the work that it does every day. On this slide, you're looking at an image taken with an electron microscope. The image has been colorized so that we can see different components of the cytoskeleton in one place. When you look at this, it just looks like a big tangle of structures, but these structures are all interconnected with each other. What you're looking at in blue here, this is a microtubule. What you're looking at in green, this happens to be an intermediate filament. The microfilaments are also present in the cell at any one time. It's a very dynamic structure. It's constantly changing. It's constantly moving in order to meet the needs of the cell. Finally, on this last slide, I've included a table for you that comes out of a textbook. I like this table because it puts together some of the things that we've talked about today with regards to the three components of the cytoskeleton. First of all, it shows us the physical structure of each component, the microtubule, the intermediate filament, and the microfilament. It talks about the subunit of each one of those and some of the functions that they perform. So, a very dynamic and very critical component of any living eukaryotic cell, the cytoskeleton.